Good morning to everyone. Today we are now learn to cell cycle and cell division. First of all, what is cell? A cell is the structural and fundamental unit of life. Cells are the building blocks of all organisms. The cell was first discovered by Robert Hooke in 1665. The cells are made up of some macromolecules such as carbohydrates, lipids, protein and nucleic acids. Next, how many number of cells in organism? There are two types of organism. One unicellular organism, number two multicellular organisms. Unicellular organism means only one cell contained organism is called unicellular organisms. More than one cell contained multicellular organisms. Next, how made up of cells? Macromolecules. Macromolecules such as carbohydrates, lipids, protein and nucleic acids is a larger size. So some macromolecules together form a cells. The more than cells together formed by a tissues. The tissues are together formed by a organs. The organs are together formed by a organ systems. The whole organ system is called living organisms. Okay, next one, cell divisions. What is cell division? When a parent cells divide into two or more cells, which is called daughter cells, cell division takes places. That means one parent cells are divided into two daughter cells is called cell divisions. Next, it generally occurs as a part of a larger cell cycle. All the cell reproduces by splitting into two, wherein each parent cell gives rise to two daughter cells. Okay, next, cell cycle. The cell cycle was discovered by Prevost and Dumas in 1824 while studying the cleavage of zygote of frog. Cell growth, DNA replication and cell division constitute a cell cycle. Events happening in a cell cycle is genetically controlled. During of a cell cycle varies in different organisms and different types of cells. The average of duration human cell cycle is 24 hours, whereas yeast cell completes one cell cycle in 90 minutes. Okay. Next, phases of cell cycle. There are two phases of cell cycle, namely first one interphase, second one M phase. Interphase is the cell growth and replication of DNA. It consists of 95% of the duration of cell cycle. Next one, M phase. M phase means mitosis. Interphase is further divided into three phases. First one, G1, S or G2. G1 phase means G means cap. G1 phase or cap phase. It is the gap between previous mitosis and DNA replication of the next cell cycle. So next, S phase or synthesis phase. In this phase, DNA replication takes place. DNA content of the doubles and centrioles duplicates. It is important note that the chromosome number remains the same. Next, G2 phase or gap 2. This phase is cell continues to growth and prepare for mitosis. Okay, this is protein synthesis takes place. In an adult human being, there are many cells who do not divide. Example, heart cells divide occasionally only to replace injured and dead cells. Okay, next, these cells enter an inactive phase called G0 or quiescent stage of the cell cycle. Next, M phase. M phase or mitotic phase is the phase where the actual cell division occurs. It consists of two processes. First thing, karyokinesis. Second thing, cytokinesis. Karyokinesis means where the division of nucleus occurs. Cytokinesis things where the division of cytoplasm takes place. Okay. Mitosis. Mitosis is a process where in a parent cell divides to form two identical daughter cells. Mitosis is called equential division because the two daughter cells have the same number of chromosomes as those present in the parent cell. Karyokinesis occurs in the four sequential stages. First one, prophase, second one, metaphase, third one, anaphase, fourth one, telophase. First one, prophase, is the first stage of mitosis. Chromosome untangled and condension Condensation. Next, two chromatids attached to the centromere can be seen clearly. Each the duplicated centrosome radiates microtubules. The mitotic apparatus constitutes spindle fibers and asters. The disappear the Golgi bodies, nucleolus, endoplasmic reticulum and nuclear membrane is disappear. Metaphase. It is the second stage of mitosis. The nuclear envelope has completely disintegrated and chromosomes are spread throughout the cytoplasm of the cell. The metaphase chromosome is made up of two sister chromatids which are held together by the centromere. The small disc-like structures at the surface of the centromere are called kinetochores. The structure serves as the site of attachment of spindle fibers. The center of the chromosome is phase metaphase plate. This process is called concretion. Okay, next, anaphase. One of the onset of anaphase, each chromosome was arranged in the metaphase plate is placed simultaneously and the two daughter chromatids being their migration towards opposite poles. The chromosome was spindle fibers pulling the spindle fibers pulling causes the chromosomal disjunction. The chromosome was separate. Next one, telophase. This is the final stage of mitosis. Chromosomes cluster at opposite folds and decondensation. Nuclear envelope develops around each cluster of chromosomes and two daughter nuclei are formed. The nucleolus, endoplasmic reticulum and Golgi apparatus are reformed. Next, cytokinesis. Cytokinesis is the division of cytoplasm. This phase marks the end of the cell division. Cytokinesis occurs differently in animal cell and plant cells. In plant cells, cytokinesis is achieved by the formation of furrow. In plant cells, cytokinesis is achieved by the formation of the cell plates. That's in the pictures. Next, meiosis. Meiosis is also known as the reduction division. Meiosis is a double division in which a diploid cell forms four haploid cells, each having half number of the chromosomes. Okay, so next, meiosis consists of a two division, meiosis 1 and meiosis 2. Meiosis 1 is called reductional division because the chromosome number is reduced to have this process. Okay, meiosis 2 is called equational division because during this division the number of chromosome remains the same as produced at the end of meiosis 2. First one, meiosis 1. It is phase 1. It is an extended stage and further divided into various stages. First one, leptotin. Chromosome start condensation. That start the condensation of chromosome. Zygotin. During this stage, chromosomes start pairing together and this process of association is called synapsis. The start pairing is the process is called synapsis. The complex formed by a pair of synapsed homologous chromosome is called bivalent. At the fact 18, bivalent chromosome now clearly visible as tetrads. This stage is characterized by the appearance of recombination nodules, the site at which crossing over occurs between non-sister chromatids of the homologous chromosome. Crossing over is the exchange of genetic material, which is an enzyme mediator process. That the enzyme involved is called recombinase. Recombination between homologous chromosomes is completed by the end of fact 18. Next, diplotin. It is recognized by the dissolution of the synaptonymal complex and the tendency of the recombinant homologous chromosome of the bivalents to separate from each other except at the sites of crossovers. The egg-shaped structure is called chiasmata. Next, diakinesis. Nucleolus disappears, chiasmata separate and nuclear envelope also start degenerating. Final, cytokinesis. The two daughter cells are formed which are haploid. Next, metaphase 1. bivalent chromosomes align themselves on the equatorial plate. This is metaphase plate. The distribution of bivalent chromosome is at random. The microtubules of spindle bifurs from opposite poles are attached to the centromere of the chromosome facing towards it. Next, anaphase 1. The two homologous chromosomes separate from each other and start move towards the poles. Only one chromosome out of a pair reaches the pole. The number of chromosomes becomes half in the daughter cells. Telophase 1. The chromosomes reach the folds. Nucleolus and nuclear membrane reappear. It produces two daughter cells, each containing a single nucleus. Next, meiosis 2. Meiosis 2 is more like mitosis, and each haploid cells forms two more haploid cells. So, after both meiotic division, we get 4 haploid cells from one diploid cell. Prophase 2 is not long or complicated. Chromatin material becomes compact. The nucleolus and nuclear envelope disintegrate and disappear. Next, metaphase 2. Chromosomes align themselves at the equatorial plate. This is the metaphase plate. This enzyme kinetochores attached to the microtubules have been extended towards the equator by the opposite poles. Next, anaphase 2. Centromere holding the two chromatids split and allow the separation and movement of the two opposite folds. Next, telophase 2. Chromatid reach the fold and decondensation. That start decondensation. The nuclear membrane and nucleolus reappear and 4 haploid nuclei are formed. Final, cytokinesis. Each daughter cells of meiosis 1 divide to form 2 daughter cells. Hence, at the end of meiosis, a diploid cell gives rise to 4 haploid daughter cells. Final, see the picture. Prophase 2, a new spindle bifurs around the chromosome. Metaphase 2. Metaphase 2 chromosomes line at the equatorial plate. This plate is called metaphase plate. The attached the enzyme is kinetochore enzymes. Next, anaphase. Centromeres divide chromatids move to the opposite fold of the cell. Next, final stage. A nuclear envelope forms around each set of chromosomes. The cytoplasm is divided into 4 haploid cells. Okay, thank you.